see, can you all, oops, can you all see my screen? And hopefully you can see this little red laser pointer bopping around. Good. And I've got at least Bronwyn in the corner of the screen, so I can see you, but I can't see most of you. So if you're waving or yelling or screaming, you may not notice. All right, so the way I've set up the talk, I gave it this title of Keystones of the Sea, echinoderms and the role of curiosity in driving research forward. And I'll explain what all that means, but you might notice that the background is not an echinoderm. It's actually a picture of the coral reef that I took maybe 5 years ago now. This was in Australia on the Great Barrier Reef. And I take this in part because I'm going to say some, somewhat depressing things about the state of the reef later on. But this is just an example of one part of the reef where it's beautiful. And all this is living coral. You don't often see photographs of just a wall of living coral. But in this case, this was a great snorkel that I was on to collect starfish. We were collecting echinoderms and got to see just gorgeous, gorgeous reef. So I'll explain the link between echinoderms and the Great Barrier Reef in a minute. I'll also just note that, as Bronwyn said, I'm at William and Mary. I'm in Williamsburg, Virginia, in the biology department there. I've been here for 14 years now. And most of the work that I'll talk about happens outside of Virginia, but some of it relates, you'll see, to both Maryland and Virginia in terms of the species that we work on, which are local here as well as in Maine, where I'll talk about most of the work being done. And at the bottom of this slide is just my Twitter handle, AlanLabWM, and Instagram if you want to see pictures, pretty pictures of things that we work on, which is larvae rock, because I work a lot on larvae. All right, so let me see if I can get the slide to advance here. There we go. So here's the plan. I'm going to talk about the basics, and that's in quotes, and I'll explain why, of research in my lab and how undergraduates, I work mostly with undergraduate students, drive our success. And I'll use as an example of how we apply some of that, the notion of the decline of corals is a very applied problem that requires a basic solution. And I'll also talk about the role that curiosity-driven research plays in informing conservation challenges on the reef. And I'll give an example at the end of how poorly, actually, we understand the basic biology of echinoderms. And these are two of my field assistants, Elliot and Simon, who are my children, holding up some colorful echinoderms that we collected. This is Pycnopodia, these sunflower stars from the coast of Washington. So the echinoderms will play a central role in explaining some of these basic problems. So the first thing y'all need to know is that part of what got me excited about being a biologist was just being naturally curious about the world. Probably that's the reason you are all here right now, is you're curious about how the world works and you'd like to know more about it. And really, that's what drives our research. So these are all pictures of students smiling and laughing and being generally excited about whatever they're discovering. On the left, the student holding the shovel with the giant whelk just could not believe that snails, that was on the eastern shore of Virginia. So you guys will probably be, some of you, familiar with the whelks that live on the eastern shore. She couldn't believe snails got that big. Other students, like the one in the middle, couldn't believe that sea stars have more than 5 arms, and sometimes they do. Sometimes they have many more than 5 arms. There's a student all the way on the right is holding a slime star, and she's just got this great look on her face because you shake the star and it just exudes this just tremendous amounts of clear, very slippery, gelatinous sort of slime when it's disturbed. So these are the kinds of experiences that I give to the students to get them hooked on and thinking, hey, how does this work? So these are the questions we start with. Why are there so many different kinds of animals, and how do they work? How do any of us work? So that's when I say basic in quotes, that's what I mean by we study basic biology. It doesn't necessarily mean it's simple or easy. It really means that we're trying to discover new knowledge and information without really worrying about how it's going to be created or used. So this is sort of one end of a spectrum of research. Compared that to the other end where you have applied science. You take information that already exists and apply it to the solution of an existing problem. So sometimes the way I describe it, there's a lot of folks at our graduate school of marine science here who are interested in knowing, how many blue crabs are in Chesapeake Bay, and they want to know that, as you all know well, for understanding how much of a harvest you can have in any given year, and that's wonderful, important work. But we're really interested in, if you were going to talk about crabs, like how does a crab work? How does it survive wild swings in salinity? And that's also very important. So you need to have both the basic and the applied research. And I just want to give you this as a framework because we're really coming at some of this work from just like, how did these animals do this? Why are they so strange? And this is just a sort of little bit of inside baseball that I thought you might be interested in. Every time I submit a grant, I have to fill out a little form that looks like this, where I check, is my research basic or is it applied? So every time I do it, I don't know what they do with this information. But I find the description of it amusing. It says research undertaken primarily to acquire new knowledge without any particular application, which sort of describes what we do. But it turns out that when you do basic research and you fund basic research, you learn a ton of things that then become applied to really important problems. And so the National Science Foundation, which is what funds a lot of my work, is an organization that funds a lot of basic and applied research. But a lot of that basic research leads to things like you would never predict, like the development of vaccines, the development of the internet. All of these things that we kind of take for granted now arose from projects that were not intended to end up where they were. So this is a kind of schematic of the work we do. And I tell students that we work in sort of three subfields of biology. There's ecology, evolution, and development. And all of these areas overlap depending on the project. So sometimes we're more interested in the ecology side, looking at predator-prey dynamics. Sometimes we're looking at life history evolution and phenotypic plasticity. Sometimes we're working in a more developmental framework, eco-devo and evo-devo, which are ecological development and evolutionary development. So putting developing organisms into the context of the real world. And the projects that we do all sort of touch on all of these areas. And we do all these projects in marine invertebrates. So this talk is about echinoderms, but we work on lots of other marine invertebrates. This particular echinoderm is one of my favorites. It's a brittle star. It's my favorite animal. So this is the adult who sits on the bottom of the ocean. It looks like an echinoderm. It's got the familiar 5-part symmetry. It crawls around. It's not a sea star. It looks sort of like a sea star. It's related. It's a brittle star. And it crawls around and lives under a coral reef. So most of the time, all you'll see are the arms sticking out. You never really see the disc of the animal. And when they reproduce, they release eggs and throw them out into the water column. And those eggs get fertilized by sperm. And those eggs develop into a little larval brittle star. And the first thing you probably notice about that larva is it does not look like an echinoderm. It's bilaterally symmetrical. So it's got the same sort of symmetry that we have. You can slice it down the middle and get 2 equal halves. And it goes through this radical metamorphosis as a larva to turn into something that's got that 5-part symmetry. So the metamorphosis of echinoderms is comparable to a tadpole turning into a frog or a caterpillar metamorphosing into a butterfly, but it's actually even more extreme because they change their entire body plan from bilateral to pentameral or 5-part symmetry. So the complexity of this life cycle I'll build on during the talk. It's actually important. This is why the poll question is actually useful, because it really depends when you think about a starfish or an echinoderm. Well, are you thinking about an adult? Are you thinking about an egg? Are you thinking about a larva or a juvenile? They They look very different and they inhabit totally different parts of the ecosystem. All right, so the talk is really a tale of two stars. The local, it turns out, in Maryland and Virginia and in Maine, where a lot of the work was done, East Coast starfish, Asterias forbesi, and a starfish from very far away on the other side of the world, which is crown of thorns starfish, which is this kind of crazy, spiky looking guy here. It turns out they're both what are called keystone species. One that the work we do is in the Gulf of Maine, but it's also, like I said, in Virginia and Maryland. And the other one on the Great Barrier Reef. So a keystone species, you've probably heard that term. It gets kind of tossed around often to mean any important species in an ecosystem. It has a little bit more specialized definition to a biologist in the sense that it's an organism that if you went to, let's say you walked out not on the coral reef, but you walked out onto an oyster reef and you said, well, what's the most important animal on this oyster reef? Well, presumably you'd say it's oysters, because the oysters create the habitat that all these other organisms live in. They build the structure, and certainly those oysters are what we'd call a foundation species. They lay the foundation of the ecosystem. And the keystone species is a species that, if it may not be the most abundant, and in fact, it can't be the most abundant in the definition. So it might be somewhat rare, but if you remove it from the ecosystem, it has a whole set of knock-on effects that sort of cascade throughout the ecosystem that cause it to fall apart or to lose productivity and decline dramatically. So this gets applied to things like wolves in Yellowstone. It gets applied to lots of different kinds of animals. And it turns out that the guy who discovered this notion of a keystone species was a guy named Bob Payne, and he spent his early part of his career literally with a crowbar, a pry bar, in the intertidal of Washington, prying big starfish off of rocks and throwing them back in the ocean to show that if you removed these starfish from rocks and threw them back in the ocean, that the character of the beach, the intertidal shoreline there, changed dramatically. So he essentially did a big removal experiment. But he didn't kill the sea stars. He didn't want to kill them. He just didn't want them on the rocks. So literally just threw them in the ocean day after day after day, until he showed that the characteristics of the shoreline changed in negative ways. The diversity went way down because the sea stars eat the dominant species. And when that diversity drops, productivity drops with it across the world. We know that in terrestrial and marine and aquatic ecosystems, the lower the biodiversity, the lower the productivity of the system, and the lower the stability as well. So it's bad. And it turns out that the keystone species is often the linchpin holding that ecosystem together. And it just so happens that the original one was the starfish. So that's why I say that. This is a picture of these keystone species when they're babies. And again, this is a different larval form, but it has that same bilateral symmetry. You can cut right down the middle of it. It's got a little mouth right here. It's got an esophagus. It's got a stomach. It's got an intestine coming up here and the anus right there. And then it's got all these crazy arms coming off of it. So it looks bizarre. They swim kind of like ballerinas with these long, very, graceful movements and strokes. And they're quite beautiful animals. But again, you would not think of them as a starfish. But it turns out that life history stage is quite important to the work that we do. So I'm going to start by talking about the crown of thorns and integrate the Asterias in there. So sort of what are cots and what do they have to do with corals? Well, I'll use that term cots. It's just an abbreviation for crown of thorns sea star or crown of thorns starfish. It's actually a suite of species. It used to be thought it was just one species, but it's probably at least 4 different species. And the work that we do in Australia is focused on this Acanthaster solaris species. This is a picture of a reef with lots of healthy coral. And you see this one patch of bleached coral over here. And that bleached coral is the feeding scar of an acanthaster, of a crown of thorns. So the starfish are basically coral eating machines. They're pretty cryptic. So here's another picture that I took. You can see another feeding scar here where there's white coral. All the pink and brown And green, that's all living coral. So everything that's not white is happy, thriving coral. And then here, kind of hidden in there, is a crown of thorns starfish. And it's feeding the way that all starfish do, which is they actually take their stomachs, they stick it out through their mouths, and then they cover the coral with their stomach outside of their body and digest the coral outside of their body. If it was eating a clam or a mussel or something like that, it would actually slide its stomach inside of the shell of the clam or the mussel and digest it inside of its shell. So these crown of thorns starfish are sort of quite bad for the environment. And like I said, they're huge. So they're one can be a meter across. They're covered with venomous spines, so you can't handle them. And they're therefore very unlikely to get eaten. So they're sort of the top of this sort of, it's almost an apex predator on a coral reef ecosystem. Nothing wants to eat them. And it turns out that outbreaks, when these crown of thorns outbreak, they destroy coral reefs. And these coral reefs have been growing for, often more than a century. And they have really devastating effects. So in this case, the sea star is sort of the bad guy when he's outbreaking. I don't mean to besmirch the animals I work on, but they're not always good. And it turns out that the COTS outbreaks are occurring now with increasing frequency and intensity. And we don't quite understand why, and I'll talk a bit about that. But one of the things that certainly surprised me that I didn't understand about the Great Barrier Reef in particular is that when you think about threats to it, you probably think about climate change, global warming, coral bleaching, that kind of thing. But it turns out there are three main threats. One of them is coral bleaching brought on by a warming ocean. Another is increased storm intensity, so more cyclones that destroy the reefs physically. And the third one, which is actually responsible for the plurality of the decline, so it's the biggest factor over a 30-year period, is crown of thorns outbreaks. So about 40% of the decline is due to these starfish eating the coral reef, and much smaller proportions for the cyclones and bleaching. Now, bleaching has been getting worse as well, so I don't mean to minimize that, but it is at least comparable to the effects of these starfish. And so there's a lot of interest in Australia in particular, but all over the Indo-Pacific in figuring out how to deal with these. So here's just in that same 30-year period, coral bleaching accounted for 10% of the decline, which is sort of remarkable and certainly surprising to me. Now, these data are a little out of date, so I'm sure in the last 5 years that's changed quite a bit, because bleaching has gotten sort of more severe, but over a 30-year period, that was the case. All right, so this is a picture of a COTS spawning. So all these little spots are not speckles from poor photography, although sometimes I do take poor pictures. Those are all little eggs that come from the COTS. And it turns out that if you went out and counted up how many of them are on the reef, there are about 10 million of them. So that's a lot of starfish. And the biggest females can each produce about 100 million eggs. And that's even a little conservative. The sort of fattest female on record made 200 million eggs, which is insane. That's a lot of baby starfish at any given time. And it turns out that when you go out and you do some environmental DNA sampling, you basically go out and collect plankton samples over the top of the reef, you can find all the baby starfish. They're microscopic, so they're hard to see. But if you sample the DNA, you can find them essentially everywhere you look. And so the paper that these data come from described it as a, quote, continuous cloud of larvae, an estimated 30 billion larvae at any moment sitting in the water over top of this massive structure that we all are familiar with. And what that means is that any small change in the larval survival of these 30 billion animals that are just sitting there waiting to turn into coral eating machines can have huge effects on the COTS populations, and therefore huge effects on the coral population. So these starfish, again, are these sort of keystone species. If there's too many of them, if they get out of balance, they can devastate one of the sort of most iconic habitats on the planet, and lots of other habitats as well. So I'd argue that COTS are basically, because of this, the best studied sea stars on Earth. There's certainly more money has gone into studying them. There's probably more people studying them than any other single species of starfish, and maybe even any other species of echinoderm in general. There's just so much interest in understanding how they work. And one of the things that we did a few years ago now was we were paid by the Australian government to come and apply some of the knowledge we had to understand their reproductive biology. So this is what a larval crown of thorns looks like. You're starting to get familiar with this bilateral symmetry here. This is a normal larva here. And then this is the same animal a few seconds later. And you can see there's two of them now. There's a top part here that popped off and a bottom part down here. And the top part is just the mouth. They don't really have a brain, as you learned in the poll question also. So they don't really have a head, but this is the anterior part of the animal. This is the posterior part of the animal. So you got a mouth floating around and then a headless larva floating around, and they just split apart from one another. And it turns out that when that happens, there are now twice as many larvae as there used to be. And so what happens to the numbers, if you think about it, is if just a small percentage of those larvae, of those 30 billion larvae, did this, split into two, you could add very quickly 1 billion extra animals to this cloud waiting to develop and metamorphose and start eating the coral reef. So it doesn't take much to drive huge changes in the populations of these animals. And what this process of what's called larval cloning, where a larva splits in two, and each of those pieces is able to regrow into and regenerate into a new animal, means is that there's potentially many, many more. You're already dealing with hyperfecund animals that are producing hundreds of millions of offspring. But those hundreds of millions of offspring are themselves capable of reproducing through this cloning process. And this was something that we just had no idea existed until we wrote a paper about it in, I guess, 2019, so a few years ago now. This kind of cloning is actually really widespread, though, in sea star larvae. And it's probably best described in Asterias forbici. So if you were on the ocean in Maryland or Virginia and you found a starfish on the shore, it would probably be Asterias forbici. Here's the larval form of it again. And it turns out that they go through a very similar kind of fission process. So here's the body, and here's sort of the head, if you want to think of it that way. And the anterior portion can regrow the body, and the body can regrow the head. And some of the first sort of clear descriptions of this was from a former undergraduate of mine at William and Mary, Holly Blackburn, who went on to med school and got her MD at UVA, and then decided she was going to become a researcher studying the science of colon and rectal cancer, and is now doing her PhD on that at Yale. So she was a pretty smart cookie. But she started off studying sea star larvae, very different. And these images just show one of these, the regeneration is this is all the same animal. So this is just the top portion of that animal. It's just basically a floating mouth. The rest of the body's gone. That floating mouth eventually starts to rebuild a little bit of an esophagus, and then eventually it gets a stomach, and then eventually the stomach gets bigger. And if you start to feed it, it gets much bigger, and it becomes a normal larva, and that normal larva can go on and complete development. So for Asterias, it's actually something that just happens all the time. I was working on them this summer, and it depended on the individuals, but all of the different animals I looked at had some level of cloning, and sometimes it's low, maybe 5 or 10%. Sometimes it's high, 30, 40, 50, 60. We had a culture where all of them, literally 100% of the animals went through this process. It was very shocking. So flipping back to Australia, a graduate student of mine, most of this work was undergraduates, but this was a graduate student, Emily Richardson, who now has finished her PhD actually in Australia and is back in the US working now. And she, here she is very carefully cutting into a COTS to dissect some of the ovaries out. She and I and some colleagues from Australia demonstrated, this is one of the few data slides I'll show, so there's not much data in the top. There's lots of data I could talk about, but I'm mostly showing you pretty pictures. What we showed and what she showed was that if you increase the amount of phytoplankton food that crown of thorns larvae are given, then you increase these rates of cloning. So basically, the more food there is in the water column, the more likely they are to clone. And this makes some intuitive sense, right? If you're a small animal and the life is good and there's lots of food around, you might be more inclined to pop into pieces and have those pieces regenerate. If there's not a lot of resources available, you might be more inclined to keep all of your body parts to yourself. You need them to survive. So it didn't really matter. We did some density treatments, which is what's going across the bottom here. But basically, all the darker gray bars are the high food treatment. You can see all the darker gray bars are higher than the light gray bars. So it turned out about doubled on average the likelihood of them cloning. And one of the things, again, I didn't realize, but I learned this as I was working in Australia. When I landed in Townsville and I was taking a car up the coast to get to the island we were going to work on, I asked the driver, well, are we going to see anything interesting on this drive? It's like, I had never driven on this part of the coast before. He's like, mostly sugarcane. And I was like, okay. And it's true, in that part of the country between the ocean and the mountains, there's just a strip of land that's highly productive to grow sugarcane. So a lot of sugar for Asia comes from that strip of Australia. And it turns out that they dump lots of fertilizer, because the only thing that broke up the sugarcane fields were these parking lots. And the parking lots were just full of empty fertilizer bags. It was where they brought the fertilizer bags to be refilled that they could then put more fertilizer on the field. So it turns out that runoff from sugarcane agriculture, and these are pictures I took of going out on the boat to work on this island on the reef. We drove by this massive sugarcane factory. And this is actually a train that runs out over the water to get to deep enough water where big container ships can come and dock and load the container ships with train cars full of sugarcane. So it's a big business. And the runoff from that big business eutrophies, the fertilizer eutrophies the ocean waters, which are naturally oligotrophic. One of the reasons coral reefs are so beautiful is because they're so clear. And one of the reasons they're so clear, there's not a lot of nutrients there for algae to grow and cloud up the water. So these are animals that are used to having not very many nutrients around. But when those eutrophication events happen, rains, you get the flow of nutrients from the fields into the ocean. This sounds familiar if you live in Chesapeake Bay, and if you're familiar with all of this, it's the same kinds of processes. Those nutrients run into the ocean, that leads to phytoplankton blooms, and those phytoplankton blooms lead to more crown of thorns larvae. So this is just what I just said. You have expanding agriculture throughout the course of sort of colonized history of Australia that leads to increased fertilizer use, and that increased fertilizer use leads to increased runoff of fertilizer onto the reef. That leads to increased phytoplankton blooms. That's what the COTS larvae like to eat. When they eat it and they get happy, they clone more. And when they clone more, that also increases the numbers surviving, and the ones that are there survive better because they have more food available. So you go from a single egg to a single larva, and then that single larva can go to 2 larvae, and those 2 larvae could themselves clone. And for all we know, this goes on forever. We don't actually know if there's a limit to how often this could occur, which might explain why there's nothing but COTS larvae floating over top of the reef. This is a little bit of a subtle point, but I'll just make the point that we think larval cloning might be one reason that COTS abundance is increasing. but it's also something that they just do, even under normal conditions. So it's a trick they've had in their bag for a long time. It's just much more commonly used when the environment changes, nutrients become more abundant, and they have more food. All right, so just to bring you back and remind you about this part of the life cycle of echinoderms, these complex life cycles. This larval stage is really important. It's why my Instagram is larvae rock, because I think larvae are pretty important. And one of the things, to go back to some of the basics of biology.
John, can I, we just had a question, and Richard just said, are cots only found around the Great Barrier Reef or elsewhere in the world?
They are only found in the Indo-Pacific, but they are on reefs that are not the Great Barrier Reef in other parts of the Indo-Pacific. So yes, they're in Maria and lots of other places. So there's different species all around the Indo-Pacific. I'm kind of shifting gears, so I could take other questions if there are questions about the cots. This is not a bad time to interrupt.
No, good. Go ahead.
I'll keep going then. All right. So to get back to some sort of basic questions, that was this idea of this very applied issue in conservation, and how some of the things we were discovering led to that. These are brittle stars again. These are now in the Gulf of Maine, not in Australia. Though I did work on Australian brittle stars too. And they have this complex life cycle that I showed you. And these are just some images of the larvae. So here's a larva up at the top. And that larva will eventually metamorphose by reabsorbing all of these arms that they use for swimming and feeding up here, and starting to build a little juvenile. So here in B, this is a little juvenile that's getting built inside of these larval arms. And eventually that little juvenile will turn into something that looks like a little tiny starfish or a little tiny brittle star. It's got 5 arms and it crawls around on the bottom. And it leaves that pair of arms, that pair of larval arms behind. And that pair of larval arms can actually, just like the cots that split in half, the pair of larval arms can regenerate into another larva. And so this is, again, something that we think might kind of go on forever, that these larval stages where we think, oh, one egg becomes one larva, becomes one juvenile, becomes one adult. Well, it's not true, because one egg might become one larva that becomes a juvenile, and then that arms become another larva. And this can go on, we know, at least 3 cycles. No one's ever followed it past 3 cycles, but it can make at least 3 different larvae. And to make this clearer, because the pictures are nice, but I had a student who was a really fantastic art student. She was a biology major and art major, double major. Anna, and I wanted to feature her because I'm going to show some drawings that she did that I could not have done, and that I think really explain this nicely. So these are all her drawings. So this is the adult brittle star on the bottom. This is sort of the similar life cycle that I was describing in photos earlier: a fertilized egg, a larva metamorphosing, and then a juvenile. The sort of brown part here shows that they're benthic, and the blue part shows that they're up in the water column. And it turns out that that life cycle that I've been showing you multiple times is actually very simplified. I called it a complex life cycle, but it's more complex than that, where these arms can pop off and those arms can regenerate a new larva. And so these larval cloning events add new layers of complexity to life cycles of these echinoderms. And so the question we had was, well, what if these layers are actually even more complex than that? So we went from sort of the normal historical complex life cycle to one that was more complex. And one of the observations we made is that these brittle star larvae pop off little pieces of themselves kind of all the time. They're just dropping little bits. And most people, I think, assumed that that was just a mistake, that they didn't mean to do it. But it turned out that those little bits that they drop off, so here's just a piece of an arm. And then a few days later, it's still basically just a straight arm. But then a few days after that, that straight arm has started to regrow, and it's now building a new little mouth and an esophagus. And then it's starting to turn into a more symmetrical larva by day 12. And then by a few weeks in, it's basically, I know these sort of still are unfamiliar to you, you still have to believe me, this is basically a normal looking brittle star larva. So what that means, and I should say this was a different undergraduate who did this. This is Aaron Darby McLean, who's graduated just in 22 and is now starting grad school at VCU here in Richmond. What this means is that you had that original complex life cycle, and I said this was complex, and then I said, well, it's even more complex. And now it's even more complex because basically not only does this larva drop a pair of arms at metamorphosis, it also drops little pieces of itself sort of all along the way. And those little pieces can make new larvae. And it turns out it's even more complex than that, because some of the times at metamorphosis, they drop just one arm instead of two arms. And sometimes they resorb both of those arms. They don't always drop arm pieces. So when you start to map out all the different ways these animals can develop, in that first few slides, I showed you, well, here's this very complex life cycle, but it turns out in reality, these life cycles are what I call hyper complex. They're just incredibly complicated life cycles that we have really only begun to scratch the surface of understanding. These are just some data just to show you how often these things happen. So sometimes, this is about 25% of the time, about 30% of the time they do this. Some of these other things where they only drop one arm are rare, maybe closer to 10% of the time. So it depends on what they're doing. They don't all do it all the time, but basically all of them do it some of the time. And so, what do I take away from this? Well, brittle stars are amazing. They're my favorite animals, as I said. They're awesome. But also what we don't know about marine invertebrates, this is not just true of echinoderms, but I'd argue lots of marine invertebrates, is much, much greater than what we do know. So that motivates us. It gives us hope, but it's also a little depressing when we think about how we don't know things. Since I'm supposed to be an expert, maybe that's not what you were expecting to hear. But I think our ignorance actually gives us hope. And so how does that work? So identifying what we don't know might actually help saving coral reefs. This isn't just true in terms of understanding how COTS work and maybe understanding when and where and why they outbreak and thinking about methods for reducing outbreak, like reducing runoff into the water. But it turns out that corals themselves also clone. So these are coral babies. And you may be familiar with this notion of mass spawning events on the Great Barrier Reef. So where all of these coral species on just a few nights of the year will all spawn at once. And it's quite spectacular. I've been able to dive in the waters there while this is happening, and everything's spawning. And the water, the surface of the water actually turns red with gametes, with eggs and sperm, and the beaches in the morning are red because of the eggs and sperm that have washed up on the beach from all of these animals spawning. And the reason they do that is it's a predator satiation strategy, where basically if they all spawn at once, the fish can only eat so many corals in one night, and so many of them survive because of that. So up in the upper left here is just a bunch of coral embryos at different stages: 4-cell, 8-cell, 16-cell stages of development. And it turns out that about half of the nights when these corals spawn, it's a little bit choppy. So just picture the Chesapeake Bay when it's a little windy and you get little white caps forming. And when that happens, those coral babies, which are really fatty, float to the surface because they're fatty. And at the surface, those choppy waves rip them apart. And when they rip them apart, they don't die. They just become smaller coral embryos. So that's what's shown here: little embryos and big embryos and medium-sized embryos, and they're all different sizes. And so basically when they get ripped apart, they just become new embryos, and those embryos can make little corals. So a little embryo makes a little coral, and a bigger embryo makes a bigger coral, and here they are a couple of weeks later as little coral polyps. So this was something, there's literally the entire literature on coral cloning is one paper that's 1 page long. And I love the paper. It's a beautiful paper, but there's nothing else known about it. And it's tremendously important. So this is where I say there's a lot we don't understand about these animals and how they work. So how could you help, maybe? So this is my argument to you, is being curious yourself is the first step, and paying attention to how your backyard is changing. And I mean this literally in your yard, but also the waters that are close to your backyard. And As an example, just to keep it echinoderm centric, millions of sea stars. It was the biggest observed die-off of wildlife in history, starting in 2014. Died on the West Coast. This is an example of a sea star that died from this wasting disease, where they got these big holes just forming in their bodies. And a similar phenomenon has actually happened on the East Coast, though it's less dramatic because the sea star fauna on the East Coast is not so obvious to beachgoers. But a lot of this, we think, is driven by warming oceans. And the warming oceans actually change the biofilm, the microbes that live on the surface of these starfish. And the starfish die because the microbial films grow too quickly and they get covered because their respiratory system is basically on the surface of their body. And so the surface of their body gets covered with these thick biofilms and they can't breathe and they disintegrate and die. But the Chesapeake Bay, just to bring it home to some of you all, is warming basically twice as fast as the ocean overall, and you're gonna see changes. Now, one of the things that I would argue is that in addition to being curious yourselves, being educated and educating others is actually really critical to the science that we do. There's of course opportunities for citizen science, but just training people to be curious and look at the world and understand that there's a lot we don't know. And so these are students that I'm thinking about in my case, but it's people in general. So I'd really say, encouraging places like the Natural History Society of Maryland and others to educate people, and supporting them is really important. So I'm gonna stop there because I think it's almost 8 o'clock. I can go on, but I want to leave time for questions. I just want to say that a lot of this work that I described was funded by, as I said at the beginning, the National Science Foundation provides a lot of support for basic research. The Australian government, through Lizard Island Reef Research Foundation and the Australian Museum, funded some of this. We actually got some space funding from NASA to use satellite data to understand the phytoplankton concentrations in US waters, actually. And then places we worked, Bowdoin College in Maine, Friday Harbor Laboratories, which is part of the University of Washington, Orpheus Island in Australia, Lizard Island in Australia, and lots of undergraduate students who helped with this work, and most from William and Mary, some working in Maine, some in Australia, some in Washington, but all contributed to this. So there's other stories I could tell, and I'm happy to talk more, but I'll stop for now and stop sharing for the moment. Just stop sharing the slides, I should say, just so I can see you all and see what questions there might be.
Well, Dr. Allen, that was amazing, and we want you to keep going on because we love these stories. Kathy had a question earlier on. During the larval regeneration, do larvae just absorb nutrients from the water, or are they using stored food?
Really good question. And larvae can absorb nutrients directly from the water. So we don't know to what degree that is contributing to their regenerative abilities. In general, what we know is that the absorption of amino acids directly across the body wall from the water usually is thought to just allow them to basically keep their, essentially counterbalance their metabolic rate. So they can't grow, but they don't die. So it just keeps them from starving to death. So it probably contributes to their ability, at least early on, not to die right away. And then they're almost certainly reallocating resources from in their tissue to rebuild some of those structures that they've lost. But yeah, it's kind of amazing that they're able to directly uptake nutrients from the water without actually feeding.
Adrian asks, does the basic research have application into human stem cell research? Another
really good question. We would argue yes in the very long run. So we have a grant that we are continuing to try to get funding for, which builds on this, that includes a collaborator. In fact, I meant to put them in the talk and I just didn't have time. collaborator at Brown University who studies the very hardcore developmental biology of echinoderms, meaning how cell fates are specified at a molecular and cellular level, looking at gene expression patterns, very much a true developmental biologist. And then we have a physical biologist at the University of Pittsburgh who thinks about things like shear forces in the environment and would want to know, like, how hard does a wave have to crash before an embryo gets ripped apart? The reason I mention that in response to this question is we put together what's it's called the integrative biology program at National Science Foundation, and we're trying to get them to give us some money because we argue that this ability of echinoderms are actually quite closely related to humans if you look across all animals. And they share a lot of the same developmental traits that we do. So, for example, I'm a twin. So I'm a product of larval cloning, if you want to think of it that way. That's not the reason I study it, but that ability of echinoderms and humans and other mammals to reproduce asexually as an embryo is rather amazing, and we share it. And so understanding that ability to go from an arm to a whole nother animal, right? If you think about lizards losing their tails and regrowing their tails, this would be the lizard losing its tail and regrowing its tail, and the tail regrowing another lizard. So how could that possibly work? We don't understand it in any animal. And so we're arguing, continuing to argue with them, that that's worthy of funding. So I hope that's not something I'll do, but it's something that I hope will build the foundation for. And then, my students will maybe be the ones when they're, taking some of the newest molecular cellular techniques and making something amazing happen.
Richard asks, are sea star larvae important food for others?
Another really good question, especially when I say there's billions of them, right? That's something we are, again, I would say 10 years ago, the consensus was no, because they're chemically defended. They're packed with saponins, these chemical compounds that make them taste terrible to most fish. But there's been some recent studies in the last few years that suggest actually there are many species of reef fish in Australia that love to eat echinoderm, and particularly crown of thorns larvae. So it's an argument that people are making, again, through increased understanding of how one way you could naturally protect the reefs from crown of thorns is by improving the fish community. So if you stopped overfishing of reefs, then there would be more fish. Those fish would eat up these little microscopic echinoderm larvae, and you might have healthier coral reefs.
Adrian also asked, is there a relationship between the 5 fronds and our 5 digits? 5 has been with us a long time in evolution.
Yeah, probably not. That's probably just a coincidence. Interestingly, though, there is, in fact, I was just telling my zoology students about this, the same genes, the same bodybuilding sort of master plan genes that cause us to have arms, so not our fingers, but our arms. It's called distillus, and that distillus gene has a homologue, so same kind of gene in sea urchins, which makes all their little tube feet. So not only do they have, like a sea star, have 5 arms, but each of those arms has thousands of little feet, little tube feet that come out of them. And those little tube feet, the genes that are expressed to cause those tube feet to form, that are on the sort of the tips of their body, are the same genes that cause our arms to form in humans, or the legs of a chicken, or the flippers of the fins of a fish. They're all the same sort of shared gene family.
And well, you mentioned that we share a lot in common with echinoderms, and is that we don't have brains either? I don't know. That was just. But in terms of the regeneration, do the folks that are working with, let's say, axolotls or worms, or are the regenerative processes the same interspecially speaking, or are they specific to a species?
Yeah, another good question. Well, I'll say two things. I'm sure it is known for some of those model systems that already exist, how similar, say, a flatworm regrowing its head, like a planarian regrowing its head, is to a vertebrate growing its tail. For echinoderms, we really don't know. It's very early days, I would say, in those systems. Especially in terms of natural regeneration, most of the work that's been done, to the degree it's been investigated at that level, is where people go in and cut echinoderm larvae in half intentionally. And the species that's the best model system for that is a bat star from the West Coast. And it's the one species that we don't observe cloning naturally. But it's been selected by developmental biologists because it's easy to keep in the lab. It's like fruit flies and other animals that are model systems. They make great model systems, but they're often ecologically sort of weird. And so in this case, the animals that are cloning and falling apart just kind of on their own all the time are the species we don't know much about. And the ones we do know a lot about are the ones that seem very reluctant to clone. So we don't know. This is the short answer.
Well, it's also fascinating that you're looking at the larval, that all this is happening at the larval stage. And I was wondering, kind of going on Richard's question with the 30 billion, and they're dividing, and how do the reefs have any chance? I mean, it seems like they're outnumbered.
Yeah, well, they are outnumbered. That's true. I think the way to think about it is, from the starfish's perspective, they have to produce that many offspring because most of them will not survive. They'll get eaten. They'll eat each other. So one of the things that I didn't put it's in the PowerPoint, but I didn't have time to get to, is one of the things that we've shown in Asterias is they're super cannibalistic. So they eat each other all the time. And so the echinoderms are really a boom bust phylum, where you might have very low recruitment for many years, and then a very high recruitment event, and then low recruitment again for many years. What takes them out is disease, in the case of like the Caribbean and on the west coast of the US. If they get too abundant, there's sort of a natural control there. But there are going to be times, I think, even in natural cycles without humans present at all, where starfish or sea urchins will just come to dominate, devastate an ecosystem, and then they'll themselves become devastated by something else. And that's just the sort of, classic ecology models of predator-prey interactions. So some of it is natural, but the thing that we need to work on, meaning all humans, is when we're the ones who are responsible for it becoming out of whack. And and like I said, the runoff is what's driving the outbreaks instead of some natural cycle, because that's where the corals can't recover, because the corals are super resilient. It's actually really rewarding to me to have gone back to parts of the reef that I hadn't seen in 10 years and that I knew had bleached badly, and most of the corals had died. And there are just tons of baby corals. Baby corals are actually really cute. They're very colorful and bright, and they're obviously very small. But there are tons of them. And so there's quite a lot of reason, I'd say, to be optimistic, because corals have been around a long time. They, if we take steps to protect them, they'll be okay. It's remarkable to see how quickly it's rebounded. Parts of the reef that were, within a few years, the coral's not as large as it once was, but the coral cover is quite high. So it's lots of baby corals instead of huge, giant, ancient corals, but they're still there. So I think it's something to be. And what you see is in areas where, you know, they're protected from fishing, there are many fewer outbreaks of crown of thorns because those fish populations are there. So just having that, it sounds easy and it seems simple, but it's taken, decades of work to sort of understand the complexity of these ecological interactions. In hindsight, it seems obvious to me, but I don't think people really thought too closely about it. And there were too many variables to instantly build a model of.
And how receptive has the Australian government and the industry been to the discoveries that your team has been making in terms of runoff and amelioration?
Yeah, another good question. Like all of us who live in the Chesapeake Bay watershed, right? Like we all know that dumping fertilizer into the bay or into the ocean is not a great thing for lots of reasons. So they had already been working closely with farmers to reduce runoff. And the farmers understood and understand that, a huge portion, I mean, if you think about the Australian economy, tourism to visit the reef is a massive part, right? Like this is when most people go to Australia, that's what they want to see. The Sydney Opera House, the Great Barrier Reef. I mean, there's only, and so they understand, and they want, they don't. So I'd say quite positively. Interestingly, some of the Australian scientists don't like some of the research we've come up with because it makes their jobs harder. It makes it more complicated. So they were kind of reluctant. So we've had a little bit of back and forth in the literature, polite scientific back and forth in the literature about the importance of cloning. But I was pleased. There was a big review paper that came out a couple years ago. It was maybe 50 different Australian scientists writing about crown of thorns and understanding it. And one of the things they highlighted was our work as being important and new to the field. So I think going forward, it's going to make a difference. And it already is making a difference to the way they think about these animals, because they're, like I said, it's a pest species and they really want to control them. I think about them as being these wonderful animals I love to see more of. And in fact, I just joined the working group for the IUCN that lists threatened and endangered species, and it's all about sea stars. So we that's a new group that I'm literally going to the first meeting of in a couple of weeks. So I'm excited to see what those conservation approaches are to starfish and echinoderms.
And if nobody else has any more questions, you mentioned that the brittle star was your favorite echinoderm. Can you tell us why?
I think probably two reasons. One is sort of looking backward, which is now that I know all the amazing things they can do, I'm just like, oh my gosh, these animals can do anything. But they probably became my favorite animal because when I was a graduate student, they were what brought me to Australia. It was a project working on these animals that live very cryptically underneath the coral. So I spent, I did hundreds of dives on the reef, and what I was looking for was dead coral. So I spent all this time in like the most beautiful place you can imagine, looking for the dead parts of it. And when I found the dead parts of it, I would flip up some of the dead coral and stick my hand in, usually blindly, and hoping I didn't get like stung by a bristleworm or something, and pulling out a brittle star and bringing it back to the lab to study them. And they were quite challenging to study. They were really hard, but they were beautiful. Some of them are, just beautiful purples and oranges and yellows and just every color you can think of. And I just became enamored of them because they're really gorgeous creatures. And the larvae, I love them. They're just so beautiful, and watching them grow and develop is so cool. So I think part of it was that they brought me to a beautiful place to work on them, and I appreciated them for that. But now I'd say it's just their biology is they do so many different things. So there's so many things we could talk about that I don't have time to talk about, but they're amazing.
Well, maybe we'll have to have you come back and do a whole talk on.
Yeah, we could do more. There could be part 2 if you need it.
If anybody has any other questions, last chance for tonight with Dr. Allen. All right.
The questions you all asked were really good. Those were great questions.
Well, thank you so much for spending your time, and thank you not only for your time that you spent with us, but all of the great research that you're doing, the basic and the applied together. We really appreciate it, and we really appreciate learning what you and your students are up to, and we hope that we get to talk more and learn from you again. So everybody stay well, stay curious, stay outside, keep your eyes open, paying attention, and we'll see you soon online, in the field, or at the building. Take care, everybody.
Thanks for a great talk.
Well, you're very welcome.